introduce Elliott because, as many of you may know, Elliott was my PhD advisor. And so this is the deep introduction, and I have a lot of things, so I hope you're ready. So Elliott got his bachelor's in biochemical sciences from Harvard, and then he went on to do a PhD with Robert Baldwin, Department of Biochemistry at Stanford. And after that, he did a short postdoctoral fellowship, and he joined the Cornell faculty, where he rose to full professor and then moved to Washington University in St. Louis. So that's just a career trajectory in terms of tenure and where he has been at Cornell and in WashU, and that's where he is now, the alumni endowed professor of biochemistry and molecular biophysics at Washington University. So that's the boring stuff. Let's get to the real stuff. So Elliott is an amazing scientist who has always been at the edge, at the real frontier of where the real interesting biophysics was meeting cell biology or biological sciences. And for this reason, depending on who you ask, they would know him for different reasons. And they would think of him as really big inventor of very important things. But they would never have the full picture because he has basically moved from different fields to different fields. And it has been amazing to actually compile all these views and actually make it a timeline. So I think the real interest started by looking at conformation changes in DNA and DNA binding proteins. And that kind of led to a better understanding of fluctuations and what role fluctuations play in actual chemical reactions and what can you learn from fluctuations. So these ideas were the ones that was basically driving Elliott and the team of people at Cornell, which include also Red, into developing the technique called fluorescence correlation spectroscopy. So if you have any biophysics friends who are doing imaging and are around and you ask him, so what do you know Elliott Elson for? They say, well, he's the inventor of the FCS. And a lot of these correlation spectroscopy techniques at that time did not have the right apparatus to be really at the single molecule level, but correlations by nature are single molecule events. So when you do a correlation function analysis, you're really looking at correlation of something in itself, and you're ignoring the rest of the population. And so that technique was developed in the 70s and really had a boom, but the technology caught up with it 20 years later. And this is a signature of what Elliott does, and he does best, because he does something, and then 20 years later, there's suddenly a boom, because now suddenly we have objectives that have been completely corrected, we can actually get single molecules that go through, but the same correlation function analysis, everything that we developed is basically applied now to get this thing. So that's, if you are a fluorescence spectroscopy single molecule person, that's what you know of. If you are a cell biologist, however, you would walk in and say, well, I did this FRAP experiment. I don't know how many cell biologists are here. There's these photo bleaching experiments that are very popular nowadays with the cell biologists. And if you ask them, if you ask the people who know, they say, well, yeah, this was developed by Elson and Webb in the 70s, 80s. And these guys were actually the first people who did the original photo bleaching experiments. And they linked the photo bleaching recovery as a kind of an outer equilibrium experiment, which would complement the correlation analysis on fluctuations. So intellectually, it was all kind of completely worked out, formulated in 2 thinking papers. And then about 10, 15 years after that, Zeiss came out with the composer that could zap around the spots and look at recovery and things like that. When he moved to WashU, this is going to be long.
Are we going to give him a chance to speak?
No, that's the whole point. So when he moved to WashU, he got really interested in mechanical properties. And at that time, he had a core team with him that moved with him from Cornell, and they're Tony Price and Bill McConnell. They're still in the lab. And these core people have been really amazing in actually developing a lot of these techniques. So this one that I'm going to tell you is developed by Bill McConaughey, and at that time they were interested in forces. So now you think about biology in the 80s, you know, these guys were already interested in the fact that what happens if you push a cell? What happens? So Bill developed this thing called the cell poker, that you could actually quantitatively poke a cell and get a response, elastic response from the cell, and understand how the actin network underneath the cell actually organizes itself. This was before AFM. Okay? And these ideas were explored and part of that. And after that, it came the collaboration with Mike Sheets and understanding single particle tracking. That was the beginning of the 90s. And that ended up being a huge boost into understanding how cytoskeleton networks are dynamic, how they move, how they move around, what is the underlying mechanism by which cells move. And at that time, Elliott decided that he is done pretty much with the single cells that everybody else uses, and he started doing tissue engineering approaches. So he started assembling in the lab from chicken heart embryos, actual beating hearts, beating heart muscles that we would have in the lab beating, and we would inject different drugs at it and see what the mechanochemical responses of these beating chicken embryo hearts are in the lab. And he was basically spent another 15 years developing those biomedical engineering approaches. So with all of this said, I don't think I have done justice to this introduction, but I just tried to kind of just explain to you a little bit of the history of all the fields that Elliott has contributed. And it's my biggest pleasure to introduce him. And he has won numerous awards. He's the recipient of the Biophysical Society Fellows Award, and that was just a couple of years ago. And we are really interested in giving the talk. So thank you so much for coming.
This guy sounds really interesting. I'd like to meet him sometime. I'm going to try, in the little time that remains, to tell you two stories. One of them is this title here. We've been trying to understand nanodomain growth in artificial membranes, in lipid bilayer membranes, and I'll explain what that means. And then if there's time, I'd like to say a little bit about a new venture in the lab which has to do with trying to look at a bistable system in dipeptide transport in yeast. And I'll explain why that's interesting if I can get to that. So let's start with the nanodomain story. And this is a story that is going to say something about how we started working on this using a method that should have worked but didn't. And now we're trying to use a different method which seems to be working. So it's partly about a journey that had false starts and is now taking us to some place where we think we can get somewhere, and we have only preliminary results. Okay, so let's go back to the beginning now. What we're interested in is phase separation in lipid bilayer membranes. So now if you take a giant unilamellar vesicle, which is giant in the sense that it's about 30 microns across, that in this field is giant, and it's composed of, say, two different lipids, a high melting lipid and a low melting lipid, and you take the vesicle up to a temperature at which both lipids are in the fluid state, and then lower the temperature below the melting temperature of the high melting lipid, it will phase separate out, in this case into a solid lipid phase, embedded in a fluid lipid phase of the low melting lipid. And this vesicle has had two lipid probes put into it, lipid light molecules that have fluorophores on them, one of which partitions selectively into the fluid phase, that's this DLPC, and one of which partitions into the solid phase. And so that means that you can actually see these different phases well illustrated and at the level of light microscopy. And people have been doing these kinds of experiments now for decades to try to understand more and more about how this phase separation occurs. Now, we're interested not at the macroscopic phase that you can see by light microscopy, but we're trying to understand whether or not we can see and what the characteristics are of submicroscopic nanodomains. That is to say, we want to look, first of all, I should have said, we're using light fluctuation methods in which we put a laser beam through the membrane and then watch how molecules diffuse through that laser beam. And this just illustrates that we're sampling a very tiny fraction of the membrane with a very small optical observation region. Okay, now what do I mean by a nanodomain? I mean a cluster of lipids which is small compared to the resolution limit of the light microscope. So say something less than about 200 nanometers. Why are we interested in looking at them? Well, for a variety of reasons. Actually, the strongest motivation for me is because I want to understand something about the interactions among the lipid molecules. And by studying phase separation, we can find out information about that. But there are other reasons as well. Biologically, phase separation seemed to have an important effect on permeability of the membrane. And then the one that most people are interested in is that these nanodomains provide, liquid-ordered nanodomains provide a model for what are called lipid rafts. And lipid rafts are somewhat controversial, but probably now largely accepted entities in the membrane, which are specialized regions, phase-separated regions in the membrane, which are smaller than the optical resolution of light, smaller than, say, 100 nanometers, and which selectively incorporate lipids of specific character, cholesterol, sphingomyelins, and others, and that they provide platforms for a variety of biological phenomena. For example, internalization, virus budding, something that Saviz is very interested in, membrane, a platform for the aggregation of membrane signaling proteins, again, something that Saviz has made some contributions to using fluorescence fluctuation spectroscopy.
Are these membranes more or less single molecular layers?
Yeah, well, they're lipid bilayers.
Bilayers. Okay.
Yeah, so the polar part of the lipid is out against the, oops, what did I do? The polar part is out against the water, and then the lipid leaflets come together into a hydrophobic interior region. Okay, so let's start by thinking about it from a physical point of view. What determines the size of a stable domain? Well, it's a balance of forces, and the one that seems to have a very large effect is the line tension, which is the two-dimensional analog of a surface tension. So we have a two-dimensional specialized region of lipid, and that's surrounded by a boundary between that region and the rest of it. And if there's a mismatch now between the interior lipid molecules and the exterior lipid molecules, then that interfacial region, that line, contributes a positive free energy contribution. It destabilizes the configuration, that configuration. And so that means, if that's true, that what the lipid wants to do is to minimize the line tension. And therefore, there's a driving force that wants to increase the area at the expense of the perimeter. Just as a water droplet will grow to decrease the surface tension relative to the bulk water contained inside. So that just says what I just said. And then there's another entropic contribution which favors many small lipid domains relative to a few big ones. And so there's some kind of a balance between these. Okay, so now this just says something about how that line tension occurs. This is from a paper from the Zimmerberg lab. And the idea is that if you have typically the high melting lipids have long hydrocarbon chains and the low melting lipids have short hydrocarbon chains, and that was true in the example that I just showed you. And so there's a mismatch between these two in which at the interface the hydrocarbon chains in principle are exposed to the aqueous solvent, which is an increment in positive free energy, and then they can try to minimize this. But in any case, there's a positive free energy contribution here, and that's why you have a line tension which is going to favor large domains over small domains. Okay, so now the kinds of questions that we want to understand is, first of all, if there are these lipid rafts, and therefore nanodomains in the membrane, can they actually be stable on their own, just as lipids? Or do these rafts have to be stabilized by the incorporation of membrane proteins? And if these nanodomains actually have finite lifetimes, what are their lifetimes? Of course, there's a possibility that we start off with, as we lower the temperature, that there's a very quick transition from no lipid domains to big lipid domains. And so, do you ever see small domains? That's one of the things that we want to look at. And so, what is the distribution of the sizes of the domains if they have a finite lifetime? And how can we detect them? And then what are the mechanisms by which they grow? These are the kinds of questions that we want to ask. So here's a kinetic model, again, from the Zimmerberg lab that shows the kinds of kinetic processes that could be involved in the growth of nanodomains. So let's say we start off our experiment where we're at a high temperature, and we have a uniform or a random distribution in the fluid phase of high melting and low melting lipids, and then we make a temperature down jump, which then in effect makes the system supersaturated for the high melting lipids. They're going to want to come out of the fluid phase and come into the gel phase, and that's what's going on here until finally they've depleted the high melting lipids. They're out of the fluid phase now into these domains, and now the domains at that stage can start growing by a variety of mechanisms. One is that they can merge together. Two little ones can merge together to form a big one. Another possibility is a process called Ostwald ripening, in which large domains grow at the expense of small domains by transport of lipids out of this into that, and without any actual merger process. And then there's the possibility of the opposite process occurring, namely the budding of little domains out of big domains. We're not going to...
Nucleation problems in getting these things started?
Yeah, so this is presumably a nucleation limited process.
Is that an impurity or just...
Well, okay, yeah. So if you have these things stuck to, say, a surface, then you can get some heterogeneous nucleation. We try to avoid that. And so we hope and we think that this is homogeneous nucleation. I hesitate when I answer that question because in our own group we have questions about how much of this is simple nucleation-dependent growth and how much is spinodal decomposition. And we have lots of big fights between the theoreticians and the experimentalists about this, which are as yet unresolved. If only I understood it. So anyway, this is the kind of method that we use. This is a fluorescence correlation spectroscopy approach, and I'm going to go through it fairly quickly. So we have a microscope that illuminates. This is now for a 3D sample. Imagine that there's a single plane here, that the molecules are diffusing in the plane, and as they go through the illuminated region, which is very small, it's a diffraction limited spot of 200 to 300 nanometers in radius, in diameter, and as the molecules go through here, a pulse of fluorescent light is emitted and carried up through the microscope to a detector. And then in order to analyze this process, we have to do it statistically because, of course, say we want to know what the diffusion constant of this molecule is that's diffusing on the membrane here. Well, as one diffuses through, you get a spike of fluorescence, and you might say, well, okay, we know what the duration of that fluctuation is, and if we know the duration, we know how fast the molecule was going across, and if we know the size of the spot, then we should be able to get the diffusion coefficient from that. But in fact, that won't work because this is a stochastic process, and each fluctuation is unlike any of the other fluctuations in principle. And so therefore we have to do a statistical analysis of this, and we could do it in several different ways, but the common way of doing it in this field is to form what's called a correlation function, which basically says how fast the system loses its memory of where it used to be. And so you get this correlation function, and it turns out that for two-dimensional diffusion, the halfway point of this correlation decay looks like this. So the halfway point gives you this TD, and TD, the diffusion correlation time, is related to the diffusion coefficient in this way. So all you have to do is measure this and you immediately get the diffusion coefficient. Now, something that's actually of greater utility in what I'm going to talk about later, or right now in fact, is that if you take the normalized correlation function, because the distribution of molecules in this open system is governed by a Poisson distribution, so that the variance is actually the variance of the molecules in this open little spot is equal to the mean. That's what the Poisson distribution says. So then that means that the correlation function, this correlation function basically is related to the mean. And then if you divide it by the square of the fluorescence, then all the optical parameters come out, and then the amplitude of the correlation function is just equal to 1 over the average number of fluorescent molecules in this little spot. So to me, it's always been remarkable and gratifying that this kind of measurement gives you an absolute measure of the concentration of the fluorescent molecules in this spot. So that means now that if we take the amplitude of the correlation function and multiply it by the mean of this fluorescent record, we get what we call the brightness, which is the photons emitted per time unit per molecule. Okay, so it's effectively, that's the brightness. Okay, so now our strategy for trying to measure these nanodomains is based on this kind of an idea. We have our lipid system, we have our phase-separated nanodomains, and we have our fluorophores that selectively partition into the phase-separated nanodomain. So that means now that if we have a nanodomain of a certain size, it may have 3 or 4 or 5 lipid probe fluorescent molecules in it, and therefore, as long as there are no optical interactions among the fluorophores that either quench or enhance the fluorescence, therefore the brightness of the nanodomain that's got 5 fluorophores in it is going to be 5 times brighter than the fluorescence of the single molecules of lipid probe that are in the fluid phase. And so our whole approach then is to measure the brightness of the nanodomains, and from that, knowing something about the partition coefficient, and I'll get a little bit more on that later, we then get the size, indirectly, we get the size of the nanodomains. What I told you here just gives us the average of the number of molecules in the spot and the average fluorescence, so we just get the average brightness. What we'd really like to do is to know something about the distribution of sizes of these, and there are methods that are available to do this. I'll say a little bit about them. Some of Savise's work actually was involved in extending these approaches to two-dimensional systems in a very nice paper that he did. Okay, so this is this photon count histogram. Now the photon count histogram, imagine that we're collecting a record of fluorescent photons, and we divide our record up into time bins. And so some time bins will have 0 photons, some will have 1, some will have 10. The photon count histogram is just a plot of the frequency of having any number of photons in a time bin. So it's the probability of getting, say, 1 photon or 5 photons or however many photons in one of these time bins. That's what that is. And so then if you want to calculate it, it's very simple. You just have to have the number of photons, the probability of getting the number of photons if there are m such fluorescent molecules in the sample region times the probability of having m such molecules, and each of these is a Poisson distribution. So in fact it looks like that. I don't want to dwell on the equations, but for those of you that are interested, this is now the photon count histogram for the probability of getting n photons per time unit given a distribution of the number of molecules in the sample region. Now, I'll just mention that in order to deal with this at greater length, to sum up over many components, it's convenient to get the generating function. And if anybody wants to know more about how this all works, you could ask Savise, but I'd also be willing to answer questions. Okay, now there's a problem with this. So when we started this off, we said, okay, that's a method, it's got to work. The photon count histogram promises to give you the number of, say we've got 10 components, the photon count histogram, the math of the photon count histogram method says, okay, I will give you the relative numbers of each of those components and their brightnesses. Okay, well now as it turns out in practice, you can hardly get more than 2 components. And the reason for that is that there's a kind of degeneracy in the method that we discovered only after we went into it. This is now the chi-square of the a chi-square plot of the deviation of the dimer and monomer concentration for this model system. And it turns out that there's a kind of valley floor here. Maybe I can show it a little better here. So there's a constraint line, and all of the solutions to the problem along this constraint line are almost equally probable. So it's very, very difficult to find out what is the correct answer here. So for example, here's an example in which if we have only 10 to the 5th time bends, and that's already a lot, then the 10 most likely solutions are all almost exactly the same. Now, if we go up to 10 million time bends, we actually get the right answer. But getting 10 million time bends is not easy. It takes a long time to accumulate that data, and your sample has to be very, very stable. So this is not actually very practical. Okay, so now why does this happen? Well, one of the contributions to the ambiguity in the measurement is the fact that we and everyone else in this game use a Gaussian laser intensity profile. So that means it's got an intense region and it's got dimmer regions. And so that means that a dim molecule going through the middle of the intense region is going to give you a signal that's somewhat like an intense molecule that's going through the edge. And that level of ambiguity then doesn't mean you can't get the right answer. It just means you have to have a lot of data to get the right answer. And as a way of testing this concept, here's the photon count histogram with a Gaussian intensity profile. Here's a photon count histogram taken in which we have a beam profile which has a sharp edge and a uniform intensity. So it's a disk profile. And in that case, then, all the molecules that are in the disk have some uniform fluorescence. And if a 5-mer is in there, it has 5 times the fluorescence as a 1-mer. And so you get these peaks, and so you can immediately see that this has many more features than this has, and therefore you can fit this with better confidence limits. I mean, there are a variety of ways of looking at this, and the way we looked at it was to say, what are the confidence limits in the fitting of N1, Q1, and so on? And for the cylindrical beam, the flat, sharp-edged beam, the confidence limits are always smaller, and so that means then that if only we could get a disk-shaped beam, we would have much better results. Now, what's the problem with that? Well, the problem with that is you can get something like a disk-shaped beam, but we always want to do this experiment with a diffraction-limited spot, because if we have a big spot, it takes a long time for molecules to diffuse through the spot, and it takes us a very long time then to try to accumulate enough fluctuations to get a decent statistical accuracy. And the contradiction then is that you can't get a flat beam, as far as I know, with a diffraction-limited spot. So we're caught in this. And we have not yet done more experiments, which we intend to do, to try to see if there's a compromise region that's better, that we can get a small spot that's somewhat disk-like. And I think that would be interesting to do, but we haven't done that yet. Okay, so now another problem with this approach is that it's dependent on the partition coefficient. You have to, if you want to see what the rate is, let's suppose here, this is an example. So let's suppose now that Q is the minimum number of fluorescent molecules that allow a nanodomain to be detected in a sea of monomer molecules that are still in the fluid domain. This is the minimum detectable ratio, and it depends on the partition coefficient C, as well as the mole fraction or the number fraction of fluid phase molecules. And this is now the area of a fluorescent of the lipid molecules. And so what you see is that our measurements are dependent on the partition coefficient. And so another problem that we had was that for the systems that we were interested in investigating, the partition coefficients were never big enough for us to see very small domains. And so as a result of all these problems, after a lot of work, we decided to use a newly introduced technique which is called inverse fluorescence correlation spectroscopy. And so this is now how that works. And this was introduced into the literature a couple of years ago by Wennmalm and Widengren and co-workers in Sweden. Okay, so here's regular FCS. When a molecule diffuses through the illuminated volume, you get a positive spike in fluorescence, and you get this correlation function. Here's a PCH, which tails out to the high end because the higher brightness molecules contribute out here. Okay, now this is inverse FCS. What you do is you put a high concentration of fluorescent indicator molecules, in our case in a membrane, or it could be also in bulk solution, and the molecules that you're interested in have no fluorescence. So when they diffuse into this region, they displace fluorescent molecules from this spot, and as a result of that, the fluorescence actually goes down. So instead of getting positive spikes, you get negative spikes. And so in that case, the time correlation function looks just the same as before. The time, it turns out the formalism is just the same. But now the photon count histogram tails off to the low intensity. The bigger nanodomains have pushed this into the low intensity domain of the photon count histogram. Okay, so now how do we get the domain size? Well, I showed you before that the zero time amplitude of the correlation function for FCS gives you 1 over the number of molecules in the observation region. This is now a result that Wennmalm and Widengren and their co-workers got. And what you see now is that the zero time amplitude depends not only on the number of molecules as the FCS I should say depends, depends on 1 over the average number. This depends on the average number as well, but it also depends, as you can readily imagine, it depends on the area of the dark molecules that are diffusing through the laser beam. And so one of the big advantages of this method is that in contrast to FCS, where we have to infer domain sizes by understanding what the partition coefficient is, this actually gives us a direct measure of the domain size as long as we can get the average number of molecules. Well, now you can't get both from this one measurement, but Yanfei Zhang discovered that if you measure the skewness of the fluorescent record, then it actually gives you the average domain size, the average domain number in the limit where you have a very high concentration of the fluorescent lipid probe. So that means then that we can actually get both the average number and the domain size directly. And we don't require anything to do with the partition coefficient anymore. So this is how Yanfei does his experiments. This is an old phase diagram in the system that we're looking at, which is a distearyl phosphatidylcholine, dilauryl phosphatidylcholine mixture, and this is the mole fraction of the high melting lipid. And so he'll go to a certain lipid composition where he knows where this phase boundary is, and he'll drop the temperature here, and then he'll observe by inverse FCS how the domains is. First of all, does he see any domains, dark domains now, and and how do they evolve? And so this is an example of an experiment that he did, and what he found were that there were 2 pathways that he could observe for domain growth. One of these, and this happens very rarely, but it does happen reproducibly at very low frequency. So now what you see now are that there are negative spikes here, small negative spikes over the course of time, over the course of the first 15 minutes. And then later, there are very large positive spikes. Now, the time correlation function of the lipid molecules are about a millisecond, and the time correlation function of these is significantly longer, namely 3 or 4 milliseconds. So these are nanodomains that are diffusing a little more slowly than the monomer. And then these are big domains. And here you can see, if you look at the micrograph, these unfortunately are not very good micrographs, but if you look carefully, you don't see any dark spots here. But here you do see dark spots. And so now these big spikes correlate with the presence of dark spots in the membrane, which are now big enough to be seen by microscopy. That's larger than the resolution limit of the microscope, larger than 200 nanometers. Okay, so now this is the other pathway. You don't see anything happening for the most part, except very rarely, in this first 15 minutes. And then at 2 hours, you already see these big negative spikes, and here you see these dark spots. So in other words, what you have here is lipid molecules forming small domains which grow to big domains, and here you don't see anything until you see big domains. Now, we're not sure why this happens often, and this happens rarely, but they both do happen. So that's where this problem stands right now. And so, yeah.
This is when you mix the lipids, and that's the time zero, basically.
Well, no, the time zero is when you drop the temperature. The lipids are already mixed in the gov.
Okay.
Yeah. You drop the temperature, that initiates the process. Yeah.
Is the laser beam doing any heating?
No, it really isn't, because the amount of absorption of the excitation light is relatively small. I mean, actually, here we have a relatively high. In a normal FCS experiment, the answer is definitely not. In this experiment, we have a higher concentration of the fluid of the fluorophores, so there could be a little heating, but it's not a big effect.
Not changing the distribution?
I don't think so. We haven't done yet careful measurements where we vary the excitation intensity, but you know, one of the properties of this kind of a measurement is that if we've got such a high concentration of fluorescent lipid here, that we actually have to reduce the laser intensity, otherwise we'll blow out our photodetector. So the amount of absorbed light, even though we have a very high concentration of fluorophores, is still not that great.
So
Yeah.
one thing that occurs to me is that if you have a really bright membrane sitting in the laser beam,
Yeah.
you will be extremely sensitive to the fluctuations.
Yeah, actually, you're right, and Saviz, and Yanfei has seen that. So you have to be. That can be a very big effect, and so if he's got a preparation that are a little bit floppy, and that move up and down a lot, then he just can't look at them. So, and that does happen occasionally.
Analyze that to get the
Well, yeah, you can analyze that to get something about the mechanical properties of the membrane, and we are interested in that, but we haven't really done much with that yet. But usually he's able to do this kind of a measurement. You don't see these large kind of secular mechanical fluctuations.
So, what is the time scale that you drop the temperature? And
Yeah, that's very important. Yeah, so the temperature drop time is of the order of a minute. And so
what is the temperature? How much do you drop?
it's about 3 or 4 degrees.
Oh, that's small.
Yeah, it's a small jump. Yeah, we start off just above the phase transition temperature and end up just below it. So it's a small drop. And somebody I was talking earlier today about how heterogeneity in the lipid composition here makes our life much more difficult, because that we're not as certain of where we are in the phase diagram as we would like to be. So one of our jobs is to try to figure out how to make our GOVs more homogeneous in their lipid composition, so we can be more definite and more certain about where we are in the phase diagram. And we think that that has something to do with the fact that although we see both of these, this we see rarely and this we see often, that it's something to do with a very critical compositional property. Okay, so I think so. One of the things that we have really been interested in trying to do is to look at how the domains grow. And there are 2 ways of doing this. I want to not spend a lot of time on this so that I can get on to the other topic. But basically, one way of doing that, the way most people now do this, is to measure the diffusion time, as I described, and then relate the diffusion coefficient in the membrane to the size of the thing that's diffusing. And that's a somewhat controversial issue, is what is the dependence of the diffusion coefficient on the diameter of the entity that's diffusing in the membrane. And dating back 40 years now, there's a theory by Softman and Delbruck, and more recently there have been attempts to correct that theory with very complicated expressions for the diffusion coefficient dependence on the radius of the domain. And so that's one way. So the other way, though, is merely to use our ability to get the size and number of the domains directly from the zero-time amplitude of the correlation function for IFCS. And so what Yanfei has done is to compare the diffusion-dependent method with the diffusion-independent method. And here he sees that the diffusion-dependent method somehow doesn't show that the domains are getting bigger, whereas the IFCS method shows that they do get bigger. The amplitude method shows that they get bigger, as we observe they do. And also, in terms of the total area fraction, the diffusion-dependent method shows that there is somehow a big drop in area coverage, whereas, and this is a little uncertain, whereas this shows the diffusion-independent method, the amplitude method, shows a consistent area coverage, which is what you would expect from a system that starts off at supersaturation, comes down to a level of equilibrium between gel and liquid phases. So this is a little complicated, and we can talk about it in the discussion if we want to. And then this is something that has to do with mechanisms of phase growth, spinodal decomposition. If you look at how the radius should depend on the log of the time, log of the radius depend on the log of the time, spinodal decomposition and Ostwald ripening have small exponents, whereas the observed result was this, and a number greater than 1. And so if these things are not happening or not dominating the process that we're looking at, then maybe it's domain coalescence, which we're modeling as a first order process in the number of domains. And then if that's true, then the half time for that would be 10 minutes. So I'm putting this up just as a kind of illustration of the kind of analysis we want to do. We don't have good enough data to have any confidence in this now. But this is the way we want to proceed, is to be able to get enough good data to be able to say that we can distinguish between these. Cities.
So what's the smallest radius that you guys would able to detect?
Okay, and we're not sure about that. When I asked Yanfei, he thinks we can see somewhere between 10 and 20 nanometers. I think that's being a little optimistic, but we've never really tested it. What we plan to do now is to do reconstitution experiments in which we have a fluorescent slide, and we put dark particles on the slide of different sizes, and then scan across them and do an IFCS analysis on that to try to validate or to try to determine directly what we can see. But we haven't done that yet. We're going to be doing that soon.
Question?
That's a good question, though. Yeah.
Do you assume or do you know that if two molecules find themselves at the same phase, that they will definitely stick together on first contact?
No, we don't know anything about that. That gets to this question of the nucleation-dependent initiation of this process, and my guess is that they probably don't. Most nucleation-dependent processes require going over an activation energy barrier and probably many tries to get over it. So my guess is that they wouldn't do that. Okay, so now I still have just a little time, and I wanted to tell you about this other thing, which is a completely different story. We want to look at non-equilibrium steady states in cells. And this is another fluctuation problem. And the motivation for this problem is that imagine now that you have a cell, and the cell is in a stable state. It might be a muscle cell that remains a muscle cell, or a liver cell that does what liver cells do over a long period of time. And so it's not changing its properties very much. And therefore, all the chemical reaction systems that are in the cell are not changing their properties very much. And so that means that what they're in, each metabolic system, each signaling system is in a non-equilibrium steady state, right? It's got to be non-equilibrium. If it were equilibrium, the cell would be dead. So it's a set of non-equilibrium steady state networks. And so and so that's just what I said. And what are the what is the thing that characterizes these non-equilibrium steady states? Well, one is, let me point out a problem. You want to know what is the flux through each node of the network. But it's a steady state. Nothing is changing. So how can you tell? Well, one way to tell is by doing fluctuation spectroscopy. It turns out, and this is not something that I'll talk about unless people have questions about it, it turns out that you're going from A to B in a non-equilibrium steady state, and A has one fluorescent color and B has another fluorescent color, then it turns out you can actually directly measure the flux of A to B in a non-equilibrium steady state by doing a fluorescence cross-correlation experiment. And it turns out that if you're at equilibrium, A cross B and B cross A have to be the same by microscopic reversibility. But if you're not equilibrium, they don't have to be the same. And in fact, you can show, and if people are interested, I could point that out later, you can show that the fluorescence cross-correlation experiments directly give you the fluxes through those networks. But that's a whole different story, and I just mentioned that now. So typically, deterministic signals drive the cell from one non-equilibrium steady state to another. But if you have just a few molecules that are controlling what steady state you're in, then the fluctuations in those molecules can actually drive you from one steady state to another. And here's an illustration of that. So now let's suppose we have a macroscopic system. Actually, this is an artificial example called the Schlogel reaction. And it turns out that for a macroscopic system, if you solve these differential equations, that where you end up depends only on where you start. So here, if you start above this critical concentration, then you'll always end up here. And if you start off below, then you'll always end up there. And so where you end up depends only on where you start. But if you've got a mesoscopic system in which you have only a few molecules that determine this, then it turns out that it doesn't matter where you start at all, because you can flip back and forth between these two just from spontaneous fluctuations. So here's one steady state, here's the other steady state, and you see in this record that you just bounce up and down between them. So what we're interested in, I mean, of course, this is the basic idea of biology regulated by signaling molecules and at the macroscopic level, but what we're interested in, and other people are interested too, is whether or not there are non-equilibrium, non-linear steady states that undergo transformations of this kind. And so the system that we're looking at, so if we can find such a system, we'd like to correlate phenotypic changes, that is to say changes in the non-equilibrium steady states of a system, with fluctuations of the control molecules that govern that system. And we'd like to measure chemical fluxes through reaction networks. I'm not going to say much about that. And here's the system that we have decided, or I should say that Artem Melnikov, who's doing this work, decided to look at. And this is uptake of sharp peptides by budding yeast. Okay, so now what's interesting about this is that you have a transporter, which is called PTR2, that allows these dipeptides to come into the cell. Now, in the absence of dipeptides in the exterior of the cell, the PTR2 gene is repressed by a repressor called CUP9. Okay? Now, if there are some dipeptides or tripeptides out here, they can come into the cell. They activate a ubiquitin ligase that targets the CUP9 for degradation. When CUP9 is degraded, PTR2 is released from the repressor, and the PTR gene is released from the repressor, and PTR2 is synthesized. So there's a positive feedback loop here. The more dipeptides that come in, the more PTR2 that's synthesized, therefore the more dipeptides that come in. So this is a positive feedback system, and when you have a positive feedback system like this, then you have the possibility of what's called bistability. That is to say, a very sharp switch between the state of low production of PTR2 and the state of high production of PTR2. Okay, so a little more information about how this works, and I think I'll just skip across this because I don't have much time left. I'm going to skip across. So now you can make a mathematical model of this, and the model predicts that you should, under some circumstances, have bistability. So the question is, do you have bistability? Okay, so now here's an example of some of the first measurements that Artem did, where the PTR2 molecule was labeled with a fluorescent tag, the green fluorescent protein, and he did cell sorting. And in fact, he did indeed find bimodal character. So this is the low expressing state, this is the high expressing state, but there was a complication, and that is that the higher the density of the cells, as it got higher and higher, the low state disappeared. And in fact, without going through, I have some slides about this, but without going through it, what we found, or what Artem found, was that this was because amino acids were being produced by degradation of the dipeptides, and the amino acids then are exported into the medium, and it turns out that they also, going through here, can activate this system. So dipeptides were coming in, they were being degraded into amino acids, they were coming out, and so the whole system got driven over to the high transporter state. And so then what Artem did was to knock this system out, and then he has a much more stable. I see now that these things are not projecting very well. But anyway, it's more nearly bistable, and these are somewhat old data. I mean, they're certainly bimodal, and now he's got data that show that these are bistable over a long period of time. So we think now we have a solidly bistable system in which to look at this. Now, I wanted to draw a distinction, or a comparison between the system that Artem is looking at and a very similar system, and this is a system that's been studied now for at least 50 years. In fact, the original model for this was produced by Monod and Changeux back in the 60s, so 50 years. And the idea here is this is lactose utilization. And so just like here, you have a pump. The inducer comes in. It makes a lac repressor molecule come off the DNA. That produces more of the pump molecule, and you have a positive feedback system, just as we have here. And a very interesting paper from the lab of Sunny Shia came out not long ago, 4 or 5 years ago, in which they were able to show at the single cell level and the single molecule level how this works. And so they had GFP labeled lac permease, this pump molecule, and they could see these molecules incorporated into the membrane of the cell, one at a time. And as a result, and so now the way this system works is that the DNA has a repressor molecule that's bound to it, just as the PTR2 gene had, but this is a very peculiar kind of repressor molecule that cross-links the DNA and makes a kind of a loop. So it binds to two different regions, two different operator regions on the DNA molecule. And so what can happen is that at low, so the basic idea is that the repressor is preventing any synthesis. The activator molecule, which binds to the repressor and makes it fall off, then can liberate these operator sites and cause the production of the mRNA, which then leads to the production of the permease. Well, now, what he was able to see is that with a very low inducer concentration, he got small bursts of this permease, whereas with higher inducer concentrations, he got larger bursts of permease. And so the idea then, his interpretation, is that small bursts are obtained when one of the repressor heads is off, liberating one site, and larger bursts are obtained when both sites are freed, and that the purpose of the activator, the inducer, is just to prolong these bursts by making it harder for the repressor to come in. Well, the reason that I wanted to draw this distinction is that in this system, the main action that's going on has to do with a conformational change of the DNA molecule, of the DNA repressor complex. And that's very different from what we're looking at. I mean, the reason I bring this up is that I'm afraid somebody is going to say, oh, well, didn't you read the paper from Shia's lab? He's got it all solved. Well, our system is different from that in the following sense, that we don't have a conformation change, but rather we have a balance between the production of the CUP9 repressor molecule and its degradation. So there could be a whole variety of different ways in which in these eukaryotic cells we get these fluctuations in CUP9. One has to do with chromatin remodeling, one has to do with fluctuations of binding, and so on and so forth. I don't have time really to go into that. And so we think that this is going to be a very interesting way, if we can get it to work properly, of looking at in a eukaryotic cell, of looking at how fluctuations can drive you between two non-equilibrium steady states that are bistable. Okay, so I'll just put these out and you can look at them. And I'm only over 1 minute. So I'll take questions if anybody's interested. Oh, there's a picture.